Hello everyone and welcome to Lecture 5. Here we're going to quickly go through a few different research methods used to study brain function. We'll run through them in essentially the same order as the book with a few slight modifications, starting with ablation and then moving down to some very brief genetic methods. So when we say ablation, what we mean is damaging nervous tissue and then seeing how behavior is altered. So you can selectively damage certain areas of the brain and then note any changes in behavior in the organism. And that will allow you to estimate the function of that brain region that was destroyed. And there are many different ways that we can actually ablate tissue. Any controlled ablation is going to start with stereotaxic surgery. Because we want to make sure that we can reliably hit the same brain region in each experiment. So we use a stereotaxic apparatus to target a specific brain region. In this case, it's a fairly deep region. You can see there marked with a little red circle in the lower illustration. The top is showing you the opened head of the rat there. And you can see where the parietal and frontal plates are fusing together. That's what we call bregma. So that's where the different plates of the skull intersect the frontal and parietal. They all come together at that point, and that serves as a landmark that we can move our probe away from and thus hit the same brain region. So when looking down at the top of the skull, you simply put your stereotaxic probe down on bregma, and then using a stereotaxic atlas, something like this, you can figure out which coordinates your brain region of interest lie at. So on a real atlas, they'll actually mark units, so it'll tell you how many millimeters you need to move away from bregma, either front or back, left or right, and then how deep in order to target your area of interest. So once you identify bregma, you just simply adjust the three knobs shown in this illustration to move your probe the appropriate distance either forward or back, left or right, and then down into the brain to then make your manipulation. The manipulations vary. You can use direct electrical stimulation, in this case using radio frequency lesions. Remember that neurons are active by creating electrical currents. And so by generating electrical currents with your stereotaxic probe, you can directly stimulate neurons. You're going to stimulate any cells, any cell bodies, or any axons that are in the region of your probe. And what's done here is you generate enough of an electrical current that it hyperexcites the neurons and causes them to die. So you can do this with electricity. You can also do this chemically. Hopefully you recall that the whole purpose of generating electrical activity is to then stimulate the release of chemical transmitter. What you can do is just release chemical transmitter, release excitatory chemical transmitters. What's usually used is canic acid, also called kainate. This binds to a certain type of glutamate receptor called kainate receptors. Glutamate is an excitatory neurotransmitter, so by activating kainate receptors, you're exciting nearby cell bodies in this case, since the axons won't have kainate receptors on them. So only the neuron cell bodies in the region where you're dumping out canic acid are going to be stimulated. They'll be stimulated to the point that the neurons themselves will die. It's what we call excitotoxicity. They're excited to a toxic degree. And of course, no matter which lesion you choose, you have to do what we call a sham lesion, which is where you just simply insert the probe and don't carry out any electrical or chemical stimulation. Because there's always some mechanical damage of jamming your stereotaxic probe through the brain and, of course, drilling the hole in the skull. Any of the tissue that your probe goes through is damaged. And so you have to have the sham lesion in any of these studies that you carry out. What we're looking at on the right here is the effect of an excitotoxic lesion in the hippocampus. So you'll notice that there are some dark blue lines. Those are showing you cell bodies. So we're looking at neuronal cell bodies. The absence of blue, which is very obvious in panel B, is showing you that those neurons have died, and that's because of an excitotoxic lesion. So the probe released canic acid, it bound to its receptors, and it stimulated the neurons there until they died. Now this type of analysis is always critical. You have to have your sham lesion, but you also have to actually confirm the areas of damage. There are a few ways of doing this depending on your population. If you're not responsible for the ablation, for example, if you're studying stroke in humans, or if you're studying brain tumors, you can identify these without having to kill your subject, and that is very valuable when working with humans. Generally, people don't sign up for studies that are terminal. So you can use CT or computerized tomography. This is going to use X-rays to create little slices through the body. And you can use this anywhere. Of course, in the world of neuroscience, we use it on the head to look at the brain. And what we can see here in this brain, this is a CT scan of someone with a very a very obvious brain tumor here. You can see the large gray abnormality, which is kind of pushed around the ventricles, and that's showing you a fairly large tumor in this person's head. You could also see this by MRI. MRI uses magnetic waves to look at the internal structures of the brain by generally following the movement of water. You can also see damaged tissue with MRI. So the kind of lighter area that's highlighted with the arrow there, that's showing you fairly focal damage caused by a stroke. Of course, every stroke is different. Sometimes you have more widespread damage, as we can see here. Those lighter regions are showing us areas of tissue damage where the water can kind of move a little more freely. And because of the difference in how water moves, we can see that the tissue has been disturbed. By doing a slightly different form of imaging, we can look specifically at white tissue, at white matter. And that's what we do with diffuser tensor imaging. So with diffusion tensor imaging, or DTI, it's still MRI, but what we're looking for now specifically is the areas where the water doesn't really move very well, because that's showing us areas of white matter. That fatty myelin is going to impede the movement of water. And so in diseases where we have demyelination, you'd expect to see a reduction in your signal in DTI, and that's what we're looking at here. They just assign different colors to them to show you similar pathways. So different colors are just showing you different pathways. The color is fairly meaningless. It's just so it's easier to make sense of the picture, because you can follow the same color to follow the path. What we can see here is that the brain damaged patient here, who's in a minimally conscious state, they have fairly substantial demyelination, especially on the left hemisphere there. As such, the brain is unable to communicate with itself as well, and so the patient is unable to achieve full consciousness as a result. And then, of course, if this is an animal study and you want to be really, really sure of where your damage is, you can just use a light microscope. This has to be done post-mortem, though. You absolutely cannot image brain damage using light microscopes unless you're going to open up a hole in the skull and only look right at the surface. But if you want to see any deep brain structures, you're going to have to use a brain slice. What we can see here is what we call a crescent violet stain, which is showing us essentially cell bodies. So we can see any areas where it's very deeply purple, we have a collection of cell bodies, so we call those nuclei in the brain. There we can see a few different nuclei highlighted with those arrows. You can use a number of different stains with light microscopes. We're going to go through a few of them in this lecture. But by using a combination of different stains, you can identify specific brain structures. Most commonly what we use are antibodies to detect specific proteins, but we'll get to that in one of the later main points. So whether you damage a brain region or whether nature damages a brain region with stroke or tumor, you have to confirm where the brain damage occurred so that you can equate your change in behavior, whether it's memory loss or paralysis or whatever it may be, to that specific brain region. From there, it's helpful to then map out what other areas of the brain your identified region is connected to. And we can do that with tracing. We can trace in both directions. We can trace the efferents, the outputs, or we can trace the afferents, the inputs to your brain region of interest. We'll use different chemicals to accomplish this. So we'll start off with anterograde labeling. You can see on the bottom left of this slide, there's a little cartoon showing you how FAL works. FAL is this small molecule that's taken up into cell bodies and transported down the axons. Because it's transported down the axon from the cell body, what we're seeing is where the axons of these neurons end up. So this would be anterograde labeling because we're moving in the direction of normal neuronal communication. We receive input at the dendrites, we sum those in the cell body, and then we send our action potentials down the axon to release neurotransmitter. If you want to see this in real life, that data is shown on the right. And we'll look in the top two panels here. What we're looking at are the connections between the primary somatosensory and motor cortices. Okay, so SSP is the primary somatosensory cortex, and the MOP is the primary motor cortex. Now on the top, they're showing us FAL in green. So the green signal here is that anterograde label. They've injected in the primary somatosensory cortex. That's why it's lit up very, very brightly in green. When they go to a different brain slice, they can see that green also appears in the primary motor cortex. This tells us that the neurons in the primary somatosensory cortex took up FAL, transported it down their axons, and those axons ended up in the primary motor cortex. In order to confirm this, you should always label in both directions. So you can also label retrogradely, where you inject some compound, in this case fluoro gold, that gets taken up into axon terminals and then transported back to the cell body. So in the bottom two panels, J and K, we can see that retrograde labeling. Instead of injecting in the somatosensory cortex, this time they injected fluoro gold into the primary motor cortex, as we can see in panel K. That very, very bright kind of orangish, pinkish label there. That's the injection site for fluoro gold. If you look in panel J to the left of it, you can see that the primary somatosensory cortex is also lighting up orange. So this is telling us whether we label anterogradely or retrogradely, we can see that there's a very clear connection between the primary somatosensory and motor cortices. And this is what you would do with any brain region of interest if you're trying to figure out the neural circuitry involved in whatever behavior you're whatever behavior you're studying. Jesus. First, what you'll do is damage the brain region that you care about, look for a change in behavior, then you'll map out the connections between your region of interest and the other areas of the brain to try to explain how it's involved in the behavior of interest. We'll see neural circuits and how they affect different behaviors throughout the later parts of this class, and these techniques are going to come up again. So make sure you have a good understanding of how we map out neural circuits. Okay, once you've identified your regions of interest, you can measure and control the activity of those nerve cells. Okay, so obviously we can directly stimulate neurons electrically or chemically. We've already gone over that. You can also measure the electrical activity of neurons. What we're looking at in this illustration would be surface recordings from a multi-electrode electroencephalogram, or EEG. So what they do here is just attach electrodes to the surface of the head. Sometimes it'll go through hair, sometimes it'll be skin. contact if the person's bald, but you're going through at least skin, muscle, skull, membranes, fluid, and then you're sensing the electrical activity. So these are very, very tiny fluctuations in electrical signals, and you're measuring the electrical activity of many, many neurons all at once. So the electrodes on an EEG are very much like the macro electrodes that can be implanted. In animals, we can actually implant our electrodes under the skull into the brain. Humans, of course, usually don't sign up for that, so we use EEG for humans. But with animals, we have more options. We can implant those electrodes and measure the activity of many neurons, is what we call a field recording. Or we can use more refined microelectrodes and actually record the activity of individual neurons. Remember, when a neuron's active, it generates an action potential, and that's nothing more than electrical activity. So those electrodes are going to pick up on that electrical activity either in individual neurons or in many, many neurons. So we can measure electrical activity that way. We can estimate activity in the brain through other methods because all that electrical activity, it's not free. It costs the neurons, in this case ATP, which of course they get from glucose and oxygen. So active neurons are going to require a lot more glucose and oxygen than inactive neurons. And based on this principle, we can use a couple different ways of estimating neuronal activity. So what we're looking at here would be 2-deoxyglucose autoradiography. So 2-deoxyglucose is a modified version of glucose, and it's modified in such a way that we can't digest it. So it's not going to get completely broken down. Once it's taken into a cell, it is modified, but then it remains in the cell. So it's not going to get broken down into carbon dioxide like normal glucose would. It's also radioactive, which means that any area of the brain that picks up 2-DG is going to become radioactive. And then we can measure this radioactivity either with PET, which is another form of brain imaging, or we can simply take the brain out and look for radiation, and that's what we're seeing in this autoradiograph. So here's a couple of different brain slices. One's from an awake mouse, well, a mouse that used to be awake, and then a mouse that was under anesthesia with ketamine. And what we can see is that the anesthetized mouse brain is using a lot less glucose, which makes sense because of the anesthesia. Brain activity was reduced, and as such, glucose demands are reduced. You can use 2-DG, either PET or autoradiography, to thus look at which regions of the brain are most active during some task. Alternatively, instead of injecting this radioactive variant of glucose, you can just simply look at the change in blood flow and the change of blood oxygenation. And that's what bold imaging does. This is a variant of MRI that's going to actually estimate blood oxygenation. Since we're going to be using up oxygen as we generate that ATP that we need to keep our neurons going, areas that are highly active are going to get a whole lot more blood flow and are going to be using a lot more oxygen, and we can detect this with bold imaging. And then we can estimate the degree of neuronal activity. What we're looking at here is bold imaging showing you the amount of activity in the amygdala. So the amygdala is kind of highlighted in the upper image, and the amount of red and yellow signal is typically how they color code activity, where red has some activity, and as you move toward yellow and white, it's becoming more and more active. So by measuring the amount of amygdala activity with and without SSRIs, this can help us understand their mechanism of action. SSRIs are drugs that are taken to help reduce the symptoms of depression. And so they thus reduce negative emotionality. What we can see On the bottom is a plot for the amount of amygdala activity when shown different pictures, either fearful pictures, happy pictures, or neutral pictures. And we can see that the amygdala becomes active when patients are shown fearful pictures. So the bold signal is going to increase when they're shown fearful pictures unless they're taking the SSRI there. So you can see the blue bars are showing you the amount of amygdala activation when shown fearful, happy, or neutral pictures in the presence of an SSRI. So SSRIs are going to reduce negative emotionality, most likely, by reducing activity in the amygdala. Right? That's just what this one little piece of data is showing us. Right? So we can actually estimate the amount of activity by just simply gauging the amount of metabolism that's going on, because a more active brain region is going to undergo higher levels of metabolism. Another estimate for brain activity would be the expression of what we call immediate early genes. And they really are what they sound like. They're expressed immediately following an increase in neuronal activity. Now, immediate early genes are, for the most part, going to be transcription factors that control the expression of a whole bunch of other genes. So while the immediate early gene won't really do anything itself, it's going to regulate a number of other genes that will then go on and perform some important function for the neuron. So the immediate early gene that we need to care about would be FOS. CFOS is what we call the mRNA, FOS is what we call the protein. In response to neuronal activity, there's generally going to be an increase in calcium, which then activates different transcription factors that then lead to the expression of FOS. Okay, we won't go over how this works, but we should appreciate that when a neuron becomes activated, it's going to express higher levels of FOS so that it can then modify its function to keep up with this increase in neuronal activity. So if you want to see what brain regions are most highly active, you can kill the animal, fix the brain, slice it, and then in this case add an antibody against FOS and look for FOS levels. Here's an example of a measurement of CFOS in different aged rats. So we have adult and then elderly rats. The adult rats are shown in the black bars in panel E, and the aged rats, their data are shown in the open or white bars. We're looking at the amount of FOS expression in the cervical and lumbar regions of the spinal cord, and then they further subdivided into the dorsal and ventral horns. So we should remember that the ventral horn is the motor output, the dorsal horn is receiving the somatosensory input. And by comparing the basal level of FOS, what they show here is that neurons in the aged animals show a higher level of activity than the younger animals. And that's simply all that they can say at this point. This isn't as elegant as the previous methods, either the 2DG imaging or bold imaging, because this requires that you kill the animal. So you really only get a single time point here. But this is another way that people will use to measure neuronal activity. So neurons that are active should have higher levels of FOS, which we can detect using light microscopy. And we can also control the activity of neurons. You can do this with electrical or chemical stimulation using the exact same method that you would do to kill neurons. All you do is turn down the stimulus a bit. So you don't apply as strong of an electrical current, and you don't apply as high of levels as you would with a excitotoxic lesion. So lower concentrations of canic acid, lower levels of electrical activity. It's still going to be enough to stimulate neurons, but not enough to kill them. A more recent advance would be optogenetics. This involves expressing a light-sensitive ion channel in whatever cell you want to study. If you want to put it into a dopamine neuron, that's very simple to do. You just drive channelrhodopsin, CHR2 is the abbreviation. You drive channelrhodopsin expression under the tyrosine hydroxylase promoter, for example. The details don't matter. What we need to appreciate here is that we can put these light-sensitive ion channels into neurons, and then we can either shine them with a blue light if it's CHR2 there, or a yellow light if it's NPHR. Okay, these are two different light-sensitive ion channels. Channelrhodopsin there is activated by blue light, and that's going to be a cation channel that causes depolarization. NPHR, which is activated by yellow light, will cause hyperpolarization. Okay, so blue excites, yellow inhibits. And you can express both of these channels in the same neuron, allowing you to turn it on or off whenever you please. So what we're looking at here, those little blue arrows are showing you when they flashed the blue light. And it really does work like this. It's very, very simple to do. You just need a couple of milliseconds of blue light and you'll stimulate your neuron to fire an action potential. You'll notice in the region where the yellow light was also on, in this case it was on for the entire duration of that yellow box, the blue light, of course, isn't able to excite the neuron and stimulate an action potential because of the inhibition from NPHR. So optogenetics is a very nice way of having exquisite temporal control over the activity of nerve cells. But it requires that you actually induce the expression of these ion channels, which isn't feasible if you're working with humans. So instead, if you're working with people, what you have to rely on is transcranial magnetic stimulation. In TMS, we're using magnetic waves to essentially accomplish what we would do if we implanted an electrode. There's going to be a couple of different sources that generate magnetic waves, and they will meet at a certain point under the skull and then generate electrical activity to either activate or inhibit neurons. And you have a little bit of wiggle room. You got a couple of centimeters to work with, but for the most part, TMS can only stimulate the surface of the brain. Unlike optogenetics, where you can just implant a fiber optic cable and directly control the activity of neurons anywhere you want, TMS can only stimulate the surface. But when you're working with people, you have to try to use non-invasive measures wherever possible, and TMS is pretty nifty. Here we're going to see the direct stimulation of the hand and arm region of the primary motor cortex, and you'll see what happens here. All right. Now, if you have more of an interest in specific receptors, or if you want to identify the location of neurons that express a certain gene, you can use neurochemical methods to identify those. Okay, we've mentioned immunofluorescence a couple of times. Let's go through what it actually entails. You can detect the majority of the proteins that we're aware of using immunofluorescence. You can detect any protein as long as we have a good specific antibody against it. And in general, there's going to be two antibodies involved. It's not always the case. Sometimes you have direct immunofluorescence where the same antibody that identifies your target actually has some sort of marker, like a fluorescent tag or something like that. And that's what we see on the left side of this illustration. So the little gray circle on the bottom, that's your protein of interest, whatever it may be. That yellow antibody shown on top of it would be what we call the primary antibody because it directly binds the protein of interest. And then the little green circles might be some fluorescent molecule, where when you shine a blue light on it, it emits green light. And then you can use some fancy filters to detect green light, which shows you where your fluorophore was, which shows you where your antibody was, which shows you where your protein of interest was. Generally, the direct immunofluorescence is going to have a much lower signal than using indirect immunofluorescence, where you use a secondary antibody. Because each secondary antibody, because each primary antibody can be bound by multiple secondary antibodies, the use of secondary antibodies is going to provide amplification for your signal. Not only that, it also makes research a little cheaper because you don't have to have a whole bunch of specially designed primary antibodies. You can just get a bunch of cheap primaries and get a few specialty secondaries. So this works pretty much the same way. You have your target of interest. That's the little gray circle. Now the yellow primary antibody you can see has no tags on it. So if we just applied the primary, we wouldn't see anything. After we apply our primary in indirect immunofluorescence, we then apply a secondary antibody that has some kind of a marker. It could be a fluorescent dye, it could be radioactivity, it could be some enzyme that will carry out a reaction. It totally depends on what setup you use and what your assay is. For this, we'll think of it as a fluorescent marker. And what this looks like whenever you then take it to the microscope is something very much like this. Here's the results from some immunofluorescence experiments. So the top two panels there, A and C, at the very top we're looking at TH, tyrosine hydroxylase. This is the enzyme required to synthesize dopamine. You can tell from the label that's going to look red, and if you go down to the third row you can see the tyrosine hydroxylase signal shown in red. Now by using different fluorescent molecules you get different colors. And so the nifty thing about immunofluorescence and light microscopy is that you can look at the presence of multiple proteins simultaneously. So not only are we imaging tyrosine hydroxylase, we're also looking at the expression of GAD. And GAD is the GABA synthesizing enzyme. And what we can see here is that there's really no overlap between our TH and our GAD signal, telling us that the neurons that synthesize dopamine and the neurons that synthesize GABA are probably not the same cells. If you use a higher magnification, that's what we see in panels B and D, you can see the similar lack of overlap. There's green, there's red, there's really not both. So here we're looking at enzymes that synthesize neurotransmitter. You could just as easily stain for a dopamine receptor using immunofluorescence. Of course, you can look at receptors in live subjects as well. And that's what we're looking at here. In this PET scan, we're looking at the radioactivity given off from some sort of ligand. So in order for this experiment to take place, what you have to do is have a radioactive version of some ligand, some molecule that will bind to your receptor of interest. In this case, they have a ligand that binds to D2 dopamine receptors. It's radioactive, so wherever it sticks, it's going to emit its radiation, which can be picked up in the PET scan. And it's only going to stick to the regions that have dopamine receptors. Of course, the more receptors there are, the more ligand that can bind, and thus the more radioactivity that will be detected. And so what we're looking at here on the right, we're looking at the level of D2 receptors as estimated in a PET scan in normal individuals, shown on the left, and then folks who are addicted to cocaine, meth, alcohol, or heroin. And what we can see here is that the amount of dopamine receptors are reduced. Hopefully we have some understanding of why that happens based on the previous lecture. But that's very readily detectable here in this PET scan, and no one had to die to capture these data. You don't have the fine resolution like we do here, so we can't see individual cells like we can with immunofluorescence. And we can't look at more than one thing at a time, but we don't have to kill our subjects either. So, pros and cons. Now, you can also estimate the concentration of some chemical of interest using microdialysis. So, this involves stereotaxic surgery, just like we've already discussed, but instead of putting an electrode or something like that, you put in a cannula, this little essentially open tubing that's also going to contain some dialysis tubing, which is going to allow very small molecules to diffuse across it. And what you do is pump some fluid through your cannula into the dialysis tubing. Small molecules are going to diffuse into the dialysis tubing. Let's say you want to measure the concentration of dopamine in some brain region. You can certainly do that with microdialysis. So by perfusing this fluid, which does not contain dopamine, through your dialysis tubing, any dopamine that enters, must have come from the brain. You just simply compare that amount of dopamine to some standard curve that you generate, and you can estimate the rough concentration of dopamine. Now, a couple of drawbacks to this is that these dialysis probes are pretty huge, so you don't really get synaptic measurements, which is really the area that we care about. It's only the dopamine at the synapse that's going to bind to the most pertinent receptors. The extrasynaptic measurements can be useful, but they could also be somewhat artificial because you've generated all this tissue damage by jamming a probe into the brain. The other issue is that you have to have a good normal curve. You have to have a good standard curve to compare against, otherwise your estimates of the concentration aren't really that accurate. Okay, so all you're getting here is some amount of diffusion, which is based on concentration. Higher concentrations of dopamine are going to translate to more diffusion of dopamine into your microdialysis tubing, but you don't really get absolute measures. Any absolute measurement is really just an estimate. Okay, genes kind of matter. So there are a number of genetic methods that we use. We're not going to go through hardly any. I'm going to focus on the ones that are covered in the book here. But before I do that, just a quick reminder of the central dogma here. Hopefully we all know what gene expression is. Genes are going to be the portions of our DNA that we turn into RNA and then eventually protein to accomplish some function. That's the standard definition of a gene. Certainly there are some exceptions to this, but that's good enough for us here. Genes are the part of the DNA that get turned into RNA and then protein. And that's the central dogma for us. That's the flow of information. So if we have a mutation in our DNA that leads to a mutation in our RNA and then a mutation in the protein, well, that's going to then affect how our cells function. So genetic differences at the DNA level could very well translate into changes at the protein level. And when your proteins are different, then the cells that express those proteins are going to be modified in some way. We can estimate the genetic contribution to our behavior or any psychological area of interest using twin studies. These can be tough to do because you have to find your twins, and then there are some other criteria to keep in mind. But aside from the difficulties of this, twin studies are pretty nifty because there are two types of twins out there. There are the identical twins, the monozygotic twins, who are nature's clones. They have the exact same versions of every gene, except for the couple hundred mutations that occur during development, but at least they started off genetically identical. And then there are dizygotic twins. Those would be the paternal twins that really are just siblings who happen to be born at the exact same time. So 2 sperm, 2 egg make dizygotic twins. They could share anywhere from 0 of their genes to all of their genes could be the same version. They're probably somewhere in between. It's highly unlikely that they are clones of one another, although it is possible. It's not probable. So they're about as genetically similar as brother and sister. The ballpark estimate is 50%, but keep in mind this varies wildly. from sibling to sibling and from dizygotic twin to dizygotic twin. Okay, so by comparing then the similarity of monozygotic twins and dizygotic twins, we can estimate the degree to which your genetics matters. The example shown here is a comparison of different personality traits in monozygotic and dizygotic twins. Just looking at the similarity, we're looking at the correlation between the two twins, where one would be a perfect correlation, where they're both exactly the same. 0 would mean no correlation, and a negative 1 would mean they are absolutely opposite of one another. What we can see here is if we compare the dizygotic and monozygotic twins, as a whole, monozygotic twins tend to have a much better correlation between all the different personality traits shown here, such as social potency, achievement, social closeness, etc. These correlations tend to be much higher than that in dizygotic twins. Now, what they've also done in this study is look at twins that are raised apart and twins that are raised together. So MZA, monozygotic twins, identically on the genetic level, but the A means they're raised apart. So genetically the same, their environments are different. DZA, genetically they're different and their environments are different because they're raised apart. MZT, monozygotic, so they're genetically similar, or genetically identical. T means they are raised together. So same environment, same genetics, right? So by comparing monozygotic and dizygotic twins and twins that are raised apart and together, we can estimate the degree to which the environment and our genetics is controlling our behavior. We can, of course, sequence our genes. Now sequencing a whole genome is totally plausible. And so you can identify what mutations tend to show up in certain groups of people. For example, if we look at the data shown on top, what we're looking at is the presence of a certain mutation in an acetylcholine receptor and how this varies between people who don't smoke or people who smoke very few cigarettes to very many cigarettes. So you can see the number of cigarettes smoked per day shown on the left. It goes from low to high. And then you can see a few different variants of this gene. And alleles would be different versions of a gene. What the experimenters are trying to argue here is that the presence of the T allele is a lot more common in people who smoke more heavily. And as such, this variant of the nicotinic acetylcholine receptor, which is of course what nicotine binds to, that genetic abnormality is probably what's predisposing them to using nicotine more often. You can find this for a number of diseases. For example, you can find the mutations that are involved in Alzheimer's disease. There's about 1% of Alzheimer's cases that are going to be heritable, and we can trace those to mutations in the enzymes responsible for producing beta amyloid. So now we can identify what protein we might need to go study to understand this disease. Keep in mind, though, that these are simply correlations for the most part until you actually go do the animal studies and show causation, so there can be disagreement in the literature. The example given here would be the degree of novelty seeking. From what I can find, I see about 13 studies that are showing that a longer version of this D4 dopamine receptor is associated with higher novelty seeking. And I see 7 reports saying that it's actually shorter versions of the D4 dopamine receptor associated with higher novelty seeking. So in other words, the kind of addictive behaviors, some say are associated with long versions, some say shorter versions. So there's not really much of an agreement here. It might actually have everything to do with this receptor, but the length of the allele might not matter. But in the end, in the end, our genetics can only take us so far because our nervous system is designed to change with experience. That's how we form memories. This is the function of the nervous system, to take in environmental stimuli and then modify our behavior as a result of that. So you're never going to find any personality trait or behavior that's strictly dependent on genetics with absolutely no environmental input. Okay, so there's always some environment involved. But you can certainly use these genetic techniques to better understand brain function. Okay, you can knock out certain genes, and what we mean by that is simply remove them from the genome so they're no longer expressed. What we're looking at on the bottom there would be anxiety behaviors in GAL3 knockout mice. Okay, so a knockout mouse has one of its genes knocked out, in other words, removed from its DNA in some way. So a GAL3 knockout mouse no longer expresses GAL3, and GAL3 is just one of the neuropeptides that our nervous system will use for signaling. And what we can see here is that GAL3 is probably important in regulating anxiety. So if we just look at these data, panel A on the bottom is showing us the amount of time spent in the open arm of a maze, and panel B is showing us the amount of time spent in the closed arm. That's pretty much all we need to care about. Now, what we need to keep in mind is that rodents tend not to spend time in open areas. They usually spend more time in closed areas because they're prey rather than predators, and so they don't like to be in open areas. So a more anxious mouse should spend less time in the open areas and more time in the closed areas. Of course, there are other ways of interpreting this data, but that's the interpretation here. And so in GAL3 knockout mice, because GAL3 is normally there to kind of suppress anxiety, that's gone. So these mice become more anxious, and as a result, their behavior changes. Now, it's not the case that they just don't move around. Okay, that's what panels E and F are showing us. Instead, it's likely a preference based on their level of anxiety. Now, it's somewhat time-consuming to generate a knockout mouse, and it's expensive. A more recent way of manipulating the genetics of your experimental animals would be RNA interference. Rather than knocking out, what we're going to do is knock down the expression here. So within all of our cells, we have what we call microRNAs. These are really small little bits of double-stranded RNA that are usually generated from some kind of a hairpin. That's just where RNA folds back on itself, and then it base pairs, just like your regular old RNA and DNA base pairing that we've learned about. But RNA should be single-stranded, so any double-stranded RNA gets cut up by the cell and then used to turn off whatever parts of the DNA were producing that double-stranded RNA. Okay, you don't need to know any real details about RNA interference other than we will either inject double-stranded or what we call antisense oligonucleotides that are going to target our gene of interest and then turn it off. And the way that they turn it off is by binding to that messenger RNA, that RNA that's created from DNA that will become protein. Because the antisense RNA is complementary to that, it's going to bind, it's going to create double-stranded RNA, and the cell is going to destroy it. So when you inject antisense RNA to, let's say, the D2 dopamine receptor, that's going to cause any cell that's expressing the D2 dopamine receptor to then destroy all of the mRNA that would have been used to create new D2 dopamine receptors. And so after your antisense oligonucleotides are injected, this RNA interference shown on the right there is going to destroy any of the future products of that gene. Now you could stop injecting your antisense and expression will resume. So we haven't knocked out the gene. Instead, all we've done is knock it down. Okay, so a knockout mouse doesn't express the gene and never will. A knockdown mouse simply has the gene turned off temporarily. So it's kind of a less severe version of knockout mice. And when doing that, you avoid some of the potential issues that come up with development, perhaps relying on some protein that you care about. So if you knock it out, it kills the mouse. So you can't really study its function in adulthood. Either way, the end result is the same. That gene is no longer expressed, its protein is no longer present, and thus you can try to judge the role of that protein in some neurological behavior. Okay, that about does it for Lecture 5. Email me any questions that you have on the material, and I look forward to seeing you for Lecture 6.